Good morning. My name is Meshack Kodiko, a teacher of biology. Welcome to our today's lesson, where we're going to talk about the process of fertilization in a flowering plant. So we want to see how the process of fertilization is taking place in a flowering plant and the events that will take place after the process of fertilization here. So first of all, having known that the male gametes in the plants is what we are calling the pollen grains. So we have the pollen grains landing on the stigma. When the pollen grains land on the stigma, we know very well that the stigma cells are made up of, they are able to produce some sticky substance. This sticky substance make the pollen grains to adhere to it, or rather stick onto it. So by the time the pollen grains stick on the stigma, then they are going to withdraw nutrients from the stigma cells. So as they withdraw the nutrients from the stigma cells, you are going to find that the nutrients that are withdrawn from the stigma cells is going to enable a growth of a part that we are calling an outgrowth. The outgrowth is called a pollen tube. This pollen tube grows down the stigma, and as it grows down the style, you are going to find that it is going to be led by two types of nucleus. At the tip is what we are having, or where we are going to have what we call tube nucleus, and following behind closely is what we are calling generative nucleus. So the pollen grains, we are saying there that they absorb nutrients from the stigma cells, enabling it to develop an outgrowth that is called the pollen tube. This outgrowth here, the pollen tube then grows down the style, being led by a type of nucleus that we are calling tube nucleus. And following behind closely is what we are calling generative nucleus. So the tube nucleus will then have to enter the embryo sac through a small hole that we are calling the micropyle. A micropyle is the path through which the pollen tube is going to enter the embryo sac. And many a times we know that when you are asked to name the part of the plant where the process of fertilization takes place, it is not on the stigma as we have always known in the past. It takes place in the embryo sac. So the generative nucleus that was following behind is going to undergo a type of division, what we are calling mitotic cell division. We know very well that in mitosis, the cells divide into two daughter cells, which are always haploid in nature. So the two daughter cells here, the mitotic cell division, it is now going to form two male nuclei. I mean the nuclei, that means the plural of the nucleus. So it forms the two male nuclei. So as they both enter there, immediately the tube nucleus enters the embryo sac, it is going to disintegrate. What do we mean by the term disintegration? Disintegration means that it is going to break. And the breakdown of the tube nucleus is important because it is going to at least give a clear pathway for the passage of the two male nuclei that have been formed there. That is, the tube nucleus then disintegrates to give a clear pathway for the passage of the two male nuclei. What are these two male nuclei that are there? Those are the two male nuclei that were formed as a result of mitotic cell division of the generative nucleus. So we are now seeing that both the tube nucleus and the generative nucleus, that together with the male nucleus that have been formed here, are within the embryo sac. So while in the embryo sac, one of the male nucleus, now not the nuclei, we are talking about now a single nucleus, that is the gamete. The one of the male nucleus will fuse with a egg cell. The egg cell, when the nucleus, a male nucleus fuses with the egg cell nucleus, it forms a zygote, which is diploid in nature. While the other male nucleus will fuse with the pollen nuclei. Now, the plural, the two of them, one male nucleus fusing with the pollen nuclei to form what we are calling a triploid endosperm, or rather a primary endosperm. So the two processes here where one of the male nucleus fuses with the egg cell to form a diploid zygote, and a process where the remaining male nucleus fusing with the pollen nuclei to form a triploid endosperm, both constitute what we call double fertilization. There are a lot of processes that will occur after these processes. That is, the two double fertilization has taken place, and then after this process of fertilization here, a lot of processes or rather changes take place in the plant. That is, one, the ovules now will change into seeds. The ovary will change into a fruit. The floral parts of the flower will now wither off and become shriveled, plus other processes that are taking place there. So you can see that this process is kind of procedural, and it goes in order. So maybe just to paraphrase what we have just talked about, we've said that when pollen grains land on the stigma, they stick on the stigma. Why do they stick on the stigma? It is because the stigma cells have the ability to secrete a substance. That substance is sticky, so that it enables the pollen grains to adhere to it. Then, as they adhere to the stigma cells, the pollen grains will tend to absorb nutrients from the stigma cells. This absorption of nutrients from the cells is going to enable the growth or development of an outgrowth. That outgrowth we have called is what? A pollen tube. The pollen tube then grows down the style, being led by a tube nucleus, and then following behind is what we are calling generative nucleus. So the pollen tube then enters the embryo sac through a small hole that we are calling the what? The micropyle. Then the generative nucleus that was following behind is going to undergo a type of cell division, and the type of cell division that we are talking about here is mitotic cell division, resulting in development of two cells, which we are calling two male nuclei. We are now having two male nuclei that are entering the embryo sac. So the reason why the tube nucleus disintegrates, or rather breaks down, is important because it enables the passage. That is, it gives a clear pathway for the passage of the two male nuclei that have been formed. So while in the embryo sac, we are going to find that one of the male nucleus is going to fuse with the egg cell. When it fuses with the egg cell, it is going to form a diploid zygote. The diploid zygote that is formed there is having the same number of chromosomes as the parental form, while the other one is going to fuse with the pollen nuclei. Fusing with the pollen nuclei is going to lead to formation of what we are calling a primary endosperm, or rather a triploid endosperm. So these two processes where one of the egg cells fusing with the egg cell fusing with the one of the male nuclei, it to form a diploid zygote, and the other male nucleus fusing with the pollen nuclei to form a triploid endosperm. The two processes constitute what we call double fertilization. And I've also said that we have processes or rather changes that take place after the process of fertilization here. We have talked about things like the floral parts withering and becoming shriveled, the ovules change into seeds. We have also seen the ovary changing into a fruit and the other processes that take place. So all of these constitute the process of fertilization in a flowering plant. I want to thank you very much for listening to my lesson of today.